Hi, Robert. I see you coming on there.
Hi, Keith. How's it going?
Good, thank you. Okay, I think we'll get going here. I think we've got probably just about everybody that's going to sign in for the day. Let me start by addressing a question that arose yesterday. And then maybe after that, Katie, I see you're on there. Maybe you could talk a little bit about recorded lectures. I've had a number of emails of people asking me about those, and I thought I would defer answers to that until we got on here, because I know you're doing things with recordings. So anyway, there was a question yesterday about what our expectations are with respect to you joining us in a on these lectures in a live sense. And I'll toss my ideas out on that, and then Katie can maybe say something about it too if she wishes. Um, but we're not taking roll. We're not keeping track. We're not. I'm not going to not remember who joined us on a current basis. That said, we would very much like you to join us as scheduled um, whenever possible. It's certainly our sincere hope that you can do that in a scheduled fashion. Um, this is a two-way street, and it's not only lecturing on Katie's part and my part, but contributions that all of you folks can make to our ongoing discussion. So we just love to have you as often as possible. Hopefully the recording things will work out. And if you're not able to make it for whatever the reasons might be, so we know you've got tremendously full schedules. Um, uh, you can pick things up on a recording, and then if that still leaves some questions in mind, you can certainly pose us for questions after that. So anyway, please join us, but it's not an absolute requirement. Okay, Katie.
Okay,
You say
so
something about recordings.
yeah, so it took me a little troubleshooting to figure out between yesterday and today kind of how I wanted to do this. And what I've done thus far is to take the recording from yesterday, the second lecture, and I've uploaded it to YouTube, and then I've embedded that YouTube video into the Moodle site. So you should be able to just go to your Moodle, and if you scroll down below where the lectures are posted, then you should be able to even watch the lecture right there in an embedded video. Now, if at any point in this process you have technical difficulties or have trouble viewing videos, or for some reason it's not opening for you, feel free to email me because I'll be doing a lot of the technical posting of all of the big videos. And so if you prefer the video to be posted in another place, I did contact Jean Carter, and she said that she was creating a box file of all the recordings as well. So I might just post it in both places just to cover all of our bases and that make sure you have complete accessibility. But I just posted it on YouTube just because I know that because it's an online platform, no matter if you're a Mac person or if you're a PC person, you shouldn't have a lot of issues viewing it. So if anyone wants to give me any feedback there or prefers another method, please let me know. But I'm hoping that the embedded video on Moodle makes the most sense, because then you don't have to jump to a bunch of different places to figure out where the recording is.
So Katie, let me just ask you myself, did we get both lectures recorded yesterday or just one of them?
Just the second lecture was recorded. I recorded it onto my personal computer. The first lecture, I started recording about, I'd say 40 minutes into the cloud, but I wasn't able to locate the recording on the cloud. So right now I'm just pulling them all onto my computer. But if you did any recording of the first lecture that you have a copy of, I'd be more than happy to process that and get that posted.
Yeah, it was a little hard for me to tell. I got a bunch of emails about recordings.
Okay.
But the time frames that were listed for him confused me. I guess it was one of the complications yesterday of having two lectures going and just one. It would have been pretty clear, I suppose, if it had just been one. So I don't know why I got so many emails on just one recorded lecture. But anyway, I hit the record button here today. I don't know if it's recording or not. Let's hope that it is.
Well, I'm recording to my computer right now, and that's the method I used yesterday, and it was effective. So this should be being captured. And then maybe sometime between now and next week, you and I can sit down and just look at either your email or your computer and kind of figure out if we have a copy of that first lecture.
Okay. Yeah, let's sure hope that we do. Well,
Okay.
thanks a lot, Katie.
Yeah, no problem.
Now, I'll open it to questions that anybody might have, as we would do at the start of any lecture. And if you don't have any, that's not a problem. Again, we won't hold that against you if you don't have any questions. But certainly, always give opportunity at the beginning of the hour for questions if you happen to have them at that time. While you're thinking about that for a minute, I'll just hope that everybody's surviving these early days in the new semester. It's always a challenge making the adjustments anyway, let alone under the conditions that everything exists right now. So certainly hope that things are playing out well for you so far. Okay, I'm going to do a little current event thing. If there aren't any questions at this point, this was brought to me by a student. I appreciate very much the input, and I'll try to say something about it. And let me before I really get going on it, let me try to get our PowerPoint up here on the screen. And I'm going to, for just the moment, I'm going to shoot ahead a little bit here to right here. And a little bit later on in the lecture, we'll be talking about sources of drugs, both synthetic and natural. And so one of the slides that I like to show in that regard is this one of a cannabis greenhouse somewhere. I've kind of lost track over the years exactly where this was. I tell people.
You aren't sharing your screen.
Can you not see the screen?
No.
Can no one see it?
No, it's still a view.
I'll be darned. Were you able to see the screen yesterday?
Yes.
Yeah, it worked out yesterday. Well, let's try it again. Any thoughts, Katie, on why I'm not? On my screen it says I am screen sharing.
Yeah, so right now it's showing up as your Zoom page.
It's showing up as Zoom.
Yeah, so you just need, I think, to select the screen that has your PowerPoint.
Well, let me try again here.
So Keith, where is your PowerPoint open at?
It was open on my screen.
Oh.
Just hang on. Excuse me, everybody. Hang on for a minute and let me try to. Okay, well, let's try it again here. Let me record. Katie, any thoughts on helping me out here?
I think, can you share your screen, just what you generally see? That might be the easiest thing. I'm also opening up the presentation, and I could also do the share screening of the presentation from my computer and have you narrate. I think that's kind of the backup plan.
That'd be terrific if you could do that.
Okay, I'm opening it now.
Is it, what's it looking like, everybody?
It's still your face, Keith. Can you just hit share screen so we can see what your desktop looks like?
Here's my, the full desktop. Can you see anything there?
No.
No.
Keith,
Yes, anybody?
do you use the web browser version or do you use the actual app?
I've just been bringing it up as the actual app.
Okay. I was wondering if maybe the web browser version was a little less user friendly, but here we are.
What options does it give you when you hit share screen?
I'm not even finding that now. I've really messed things up.
Nope, we can see the slideshow now.
I just put that up because I figured it'd be easier. And it was allowing me to.
Awesome.
Okay.
So I don't know if that helps at
So
all.
is it up? Is it up
Yeah.
for now for people?
Yes.
Okay, let me get mine back.
Keith, just tell me where you want to start off.
I want to start off with that slide with the cannabis greenhouse. Yeah, that's great. Yeah, there.
Okay.
Okay. I should have just not brought that up. But anyway, I wanted to start with that because cannabis is an important topic for us, and later on in the semester, a month or two from now, we've got a segment on cannabis, which you know is an important drug topic that's been dealt with, you know, for a very long time, but in terms of sort of formal pharmaceutical science education, often it's not been a significant part of it relative to many of the other more standard drug topics. But as we all know, you know, cannabis is hugely important as far as modern culture is concerned in the United States and elsewhere. So we're trying to emphasize it more than we have in the past. The current event that was brought to my attention by another student relates to cannabis and to COVID-19 both. So it's kind of a neat current topic in that regard. I'll give you the title of this article that this information came from, and I'll just do a little bit with this. I won't take too long with it. The title is "Cannabis for COVID-19: Can Cannabinoids Quell the Cytokine Storm?" and it's published in an online journal called Future Science, July 2020. So it's very current. It's not very long. It's kind of a technical review on this subject. It's about 4 pages. And if anybody is interested enough in this, I can certainly post it up on Moodle for us. Um, I won't sort of overdo posting up things like that because I don't want to clutter things too much for you. But if I hear on any one of these topics interest from people, I'll go ahead and post things up. The authors here are E. Onavari and V. Sharma from one of the East Coast medical schools. And here's some sort of highlights that I'll give on this, and then see if you have any questions or thought on it. As I'm sure all of you know, probably better than I do, I'm certainly no expert on COVID at all, but infection with the virus occurs by what is frequently referred to as the COVID-19 spike protein. And the spike protein is sort of the mechanism by which the virus can enter cells, often first respiratory cells, but then many other cells in the body have the receptor for this called ACE2R, which occurs on human cell surfaces. Of course, once inside the cell, the virus is rapidly and profoundly replicated. And the presence of the virus triggers an immune response that results initially in the acute respiratory distress syndrome, as well as many other pieces of complicated cellular biochemistry. The virus then triggers the release of many different cytokines. As you know, the cytokine area is a very complex one, and there's a lot of different components to this response, but one sort of fairly well-known cytokine that you're familiar with is IL-6, which is part of this storm. Now, from the cannabinoid standpoint, there's quite a bit of evidence in the literature that cannabinoids are known to suppress inflammatory cytokine storms, such as we're talking about here. And some of the best information of that comes from the world of AIDS, in which there's sort of a similar process that occurs, and then treatment of that with cannabinoids, including CBD and THC, but not necessarily limited to those. And when we discuss the cannabinoids later on, we'll find out that there's many different cannabinoidal and non-cannabinoidal processes that are involved with respect to crude marijuana. So sort of where the research is at right now is that researchers are hopeful. It's more along the lines of hopeful, but it's being tested out about whether a similar situation may occur with respect to cannabinoids being able to regulate the cytokine storm in COVID-19 infection. So I thought that was kind of a neat current event that fits both our purposes in the course as well as current topics with respect to COVID. And thank the student contributor very, very much for offering this up to you. And that was timely also in terms of being able to demonstrate to you folks that it'd be wonderful to get information from you on current topics like that, just as we saw today. So I don't know if anybody has any questions or thoughts on that, but you're certainly welcome to ask those now if you have them. And as developments occur in this area as the semester goes along, we'll try to update you along the way. And if you see anything yourselves that's of an update nature, please feel free to communicate that with us.
So in that
Anybody have
article,
any? Go
in that
right
article,
ahead.
did it mention how the route of administration would affect that? Because I would assume that smoking would irritate the respiratory tract. So would it be recommended for like edibles? Would that have the same amount, or does it need to be delivered directly to the lung?
Yeah, it's a good question. I'd have to go back and check, but I don't think they addressed that in this particular article. I mean, you've certainly hit upon an important thing about cannabis pharmacology, in that administration by the respiratory route is efficient, but nevertheless, anytime something is smoked, whether it's cannabis or tobacco or otherwise, there's potential for adverse effects and irritation along those lines. So I'll see what I can find on that, because they had a nice reference list, and if nothing else, might be able to take a look at the AIDS literature and see what they say about that. Um, good question, but I'm afraid I don't have an explicit answer for you right now. But it's certainly an important one to address anytime we're talking about cannabis. All right, thanks a lot. Well, Katie, I'll go back to the beginning of the publication now.
Keith, what slide did you want to go to?
To the first one, the very first slide. And I'll just very quickly run through that a little bit until we get up to where we were yesterday.
Okay.
That's great. So just in case for people that weren't with us yesterday, we've got a potpourri of ideas relative to hopefully important concepts that are generally useful as far as pharmacology is concerned. And then a few other slides, which I just put in for the heck of it. So there's Mr. Ed again, and I think people are taking it to heart that you're welcome to ask questions anytime you want. This is Hipshot, but later on you'll see the black cat slide in future presentations. And the reason I bring that up again is I was looking at the calendar before we started today, and I realized that on Monday was Black Cat Appreciation Day for the cat aficionados out there. So I missed that, but we'll celebrate Black Cat Appreciation Day a little bit late. We talked about nictitating membrane as a model investigational system, and then just some more cute shots of life. Discuss a little bit about the idea of what is a drug, and then launched into some particular aspects of anatomy and physiology, which are particularly crucial to our efforts in our course throughout the year, including, especially later on, the pancreas. So I think we were about right here. And this schematic of the hypothalamus and pituitary is, I think, probably quite recognizable by people because of your previous studies that you've done with hormones. The hypothalamus and pituitary are such fantastic regulatory parts of our system that I thought I'd bring it up today, because not only are they critical in terms of hormonal pharmacology, which we'll tackle in next semester, but because of things like the autonomic nervous system, for example, although we tend to think of it as autonomous in terms of peripheral physiology and pharmacology, in fact, there are central regulatory aspects, the hypothalamus being one of those, which are important in terms of our overall thoughts about regulation of the autonomic nervous system, and ultimately the regulation of the system in the context of drugs that we may be utilizing in that context. So I just tossed that in to sort of tickle our memories a little bit about some important regulatory functions of the system, which are going to be critical as we play out pharmacology. And I think most of you are with us today, but I'll just reemphasize it again, and especially for those that might not have been with us yesterday, about how critical our foundations in biochemistry and physiology are, as well as even earlier sciences in terms of our understanding of drug action. Pharmacology is the science of drug action, and there's various components of pharmacology that fit into that mode. One of which is called pharmacodynamics, and then of course pharmacokinetics. And in some people's definitions, both pharmacodynamics and pharmacokinetics are parts of pharmacology. The pharmacokineticists might argue that a little bit, and argue perhaps strongly that they have a separate discipline, but it all comes together ultimately in understanding drug action. Pharmacodynamics is that part of it in which we're really looking at the effects of drugs themselves on the system, whereas pharmacokinetics is more the effects of the system on drugs. But all of that is related to drug action, and we'll be looking at all of those aspects as we go along. I'll say a little bit more about pharmacokinetics today, but certainly we won't be doing any detail whatsoever on pharmacokinetics in this course. You folks have a wonderful pharmacokinetics course that most of you are taking anyway, and you'll get all sorts of fantastic detail on pharmacokinetics in that course. But we need to work with both those together to understand drug action. So we'll be doing a little bit even in this course with pharmacokinetics. The next slide is called "To the Parasympathetic Nervous System," and I put it up mostly just to emphasize the idea that many new technologies are now available for non-invasive informational gathering as far as human or other animal brains are concerned. And here's just what's been one of my favorite slides over the years, because it's from a research paper by a former student in the laboratory who's now on the faculty at the University of Arizona. And her interests, and this is really pretty unique stuff, is in the realm of grief. And she's published some really fantastic stuff in terms of the origins of grief processes in our brains, and how accessible those processes may be to therapeutic manipulation. And here's just one of an MRI from one of her papers, and I more than anything else put it up to emphasize the idea that there's some of these marvelous non-invasive tools that we'll make reference to along the way in terms of our own analysis. But if any of you happen to be sort of interested in that grief topic, let me know, and I can email to you, or if it's broadly interesting enough to people, post some of the publications up on Moodle. Well, as far as sources of drugs are concerned, there are, of course, a wide variety of sources, both natural and synthetic, as far as drug substances are concerned. And one that I think some of you have heard me talk about before, but others, if you haven't heard from me, you may have heard through other realms. But I like to talk a little bit about natural products as sources of drugs, the historical, and we're on, for reference sake now, we're on the slide called cone snail. And as we think about natural product sources of drugs, we may think first of all about plants, which is a very reasonable assumption to make, because think very familiar drug substances like the opioids, cocaine, and many others, of course, are coming from sources, or at least originally came from sources in the plant world. But it turns out that all living forms, to one degree or another, have the potential to deliver interesting drug substances to our analysis. And one that has come into feature in the last few years is from the cone snail, which is a magnificently beautiful organism from temperate marine waters throughout the world. And the cones on these snails are perhaps the most beautiful part of it, in that they're tremendously artistic, and they're collected for their beauty. But from a biochemical pharmacological standpoint, it's the cone snail's magnificent ability to produce a wide range of bioactive substances that makes it so useful as far as the drug world is concerned. And of course, those many different substances that are produced by the cone snail are utilized biologically by the snail to protect itself, as well as to hunt. And the connection with pharmacology, toxicology, is that these toxicological substances also have the potential to some degree to be pharmacological agents. And one of the themes that we'll see in pharmacology, toxicology as we go along is that most substances, of course, can be both beneficial and adverse, depending mostly upon the concentration or dose responsive aspects of those particular substances. So we frequently talk about one of the cone snail calcium channel antagonists, which has now been approved by the FDA, and which is useful in the realm of pain relief. And we'll talk about that in some detail later in the semester when we're talking about pain, particularly in the context of the opioids. But this would be an example of a non-opioid pain reliever. If you want to have fun sometime and you have an extra moment, which you don't have many of, but you can just go to the net and in your search engine, type in cone snails and see some neat stuff, particularly videos of their ability to deliver their toxins. They have a harpoon. It actually does look like a harpoon. It's amazing. It's tethered to a sac which contains all the variety of chemical substances that they produce. And when prey or a predator is nearby, they shoot this harpoon out. Harpoon hits the unfortunate organism that's in its way, and then the toxins move through the tether into the harpoon and into the body of the prey. And just within seconds, there's dramatic biological actions, convulsions, and various things like that occur, which are testimony to the tremendous bioactivity of the snails. You may be more familiar with organisms such as spiders or snakes, which are also tremendous producers of bioactive substances. Again, some of which are useful pharmacologically as well as toxicologically. So all of those will be things that we'll be talking about as we proceed through the course over the next many months. The next slide, I just switched to one that's titled Natural Products. And this is more in the realm of the botanical sources of drug substances. In the history of pharmacology, natural products derived from plant sources has been a very important topic, because before the synthetic era, all we had basically was drug substances from the natural world. So throughout the earth, just about in every biome that you can imagine, there are plants that produce one type or another of natural product drugs. Particular botanical garden is from Italy. It's actually from the island of Sicily in an East Coast port, Catania, and they have a fantastic natural product garden there. But you can find these in many places, including University of Montana. And as some of you may already know, those beautiful plants that are growing outside the university center here in Missoula at the University of Montana are full of important natural product drugs, which can be grown in the Montana climate. And if you ever have an opportunity and haven't had a chance to do it before, to take a tour with some experts on natural product drugs, they'll guide you through that particular garden, and it's really neat stuff. So that brings us to our cannabis picture that we had that I led you into so much trouble with earlier. And again, I'm not sure where this greenhouse is, but I often tell people it may be one that I put up a long time ago from the University of Mississippi in Oxford, in which they have a fantastic collection of cannabis. And in fact, the sort of official national research repository for cannabis is at the University of Mississippi. Closer to home, St. John's wort grows in Montana and is a tremendously important natural product as far as treating depression is concerned. And so that'll be a big topic for us later on in our central nervous system pharmacology. The National Buffalo Range grows very nice St. John's wort, and it's also present in a number of other locations locally. So you can go to faraway places like the University of Mississippi or Sicily to look for natural product drugs, or you can find them close to home. Of course, in the last century or so, chemical synthesis has become a big deal as far as production of new drugs is concerned. And pictured in this slide is Sasha Shulgin, who a number of our pharmacologists and medicinal chemists, including one well familiar to a member of our class, a number of these people knew Shulgin and recognized his brilliance as far as drug synthesis is concerned. Unfortunately, Dr. Shulgin passed away a few years back, and a major loss to the world in terms of his contributions in medicinal chemistry as far as synthesis is concerned. But you're looking at a picture of Dr. Shulgin in his home laboratory, in his garage. And he had a rather unique relationship with the DEA and other regulatory bodies in terms of ability to work on a home basis like this. But I put Dr. Shulgin up because many people argue, and I think rightfully so, that he may be the greatest medicinal chemist that has ever lived. But in the hands of Dr. Shulgin and many others, of course, the synthetic world produces a tremendous array of drug substances in some of the areas that we'll be looking at later on, taking, for example, anti-anxiety drugs as a good indication of that. There are literally thousands, in some maybe tens of thousands, of versions of chemical synthetic drugs, which were originally modeled from natural products, but then the synthesis goes far from that. So we'll be talking in both of these realms of natural products and synthetics as we proceed throughout the semester. Well, this is just a stress reliever here. This is little Francis the cat, who interestingly loved to mostly drink out of a faucet, and he was getting ready to go get a drink of water here. And I just put this up in case you have any stressful moments over the next few days. Maybe you could bring this up and take a look at Francis when he was a little kitten, and maybe that will calm things a little bit for you. But of course, an important physiological point along these lines is that water, of course, is such an incredibly important um, biochemical substance that water homeostasis is a big deal in terms of our health, and is a big deal when we're not in health also. And there are many drug substances, the diuretics, which come to mind most likely, but many others which are involved in regulating water homeostasis. And those will be important topics for us as we proceed throughout the many months ahead. There are developmental aspects of drug action, and I bring this slide forward to sort of emphasize that. By developmental aspects, I mean that as we proceed from sperm and eggs to fertilized ova, to neonates, to children, to teenagers, to young adults, to middle age, and to geriatric realm, the effects of drugs on us change because our bodies change. And it sort of brings up the whole idea of setting. It's the setting through which a drug is administered, which has such dramatic impacts upon what the ultimate consequences of that drug action are. I selfishly bring up this photo because this is a over 100-year-old photo of some of our family's relatives, in which there are young babies to much older individuals in the photo. And it's just a reminder to us that the drug action has to be adjusted, dose, types of administration, and a variety of other aspects to account for differences in ourselves as we move through the years from being youngsters to being elderly. And so those developmental characteristics of drug substances are clear complications as far as pharmacokinetics and pharmacodynamics are concerned. Very interesting complications, but complications nevertheless. Just keep in mind that as we go, we should make mention at times of the differences in a drug, whether we're administering that to a teenager, let's say 15 years old, to a middle-aged adult, let's say 40. And so those matters of developmental change impact pharmacokinetics, as I just said, as well as pharmacodynamics. And so one of the things that we need to do, and we'll stop here in just a minute, but as we kick off our lectures next Tuesday at 1 o'clock, we'll talk about some of the most important characteristics of drug disposition or pharmacokinetics, often referred to as ADME, or administration and absorption, distribution, metabolism, and elimination of drugs, to put that in the context of pharmacodynamics. And then a few general things about pharmacodynamics we'll cover after that. And that will be the end of one of our first stages of this early part of the course. But Liz will be with us after that to talk about the burgeoning area of pharmacogenetics. And then once she's done that, I'll do just a little bit more on some pharmacodynamic topics before we turn the corner into the important realm of autonomic drugs and give our first look at some of the discrete drug areas. Okay, everybody, my apologies again for my mix-ups. Hopefully there won't be too many of those, but that happens to me, I guarantee you, on computers. But thanks much to all of you and Katie for your input and getting us going. Um, have some good days in between, everyone. Enjoy yourselves, stay safe, and we'll see you next Tuesday in Zoom for a continuation of our introduction to pharmacology. Take care now. See you soon.
Thank you.
Oh, you're
Thanks,
welcome.
Steve.
Thanks to all of you. Katie, are you still there?
Yes, Keith, I am.
I don't know what happened on that screen share thing. That's weird.
Do you want to try screen sharing again right now and see if we can troubleshoot it a little bit?
Yeah, sure. You know, on my screen, the little tab that said screen sharing said that I was, and I don't understand that at all. And you know, up on my screen was